From Microbe TV, this is Matters Microbial, a podcast about the wonders of microbiology, microbiologists, and microbial centrism. This episode was recorded on September 26th, 2025. Hello, Micronauts, and welcome to our Quality Quorum for today. I'm your host, Dr. Mark Martin, Associate Professor of Biology at the University of Puget Sound in Tacoma, Washington. Today is the 110th episode of Matters Microbial. Many thanks to every listener, viewer, guest, and supporter. It truly matters. As you can see here, Carol wrote this email to me asking about mycobacteria and water, which is a fascinating topic. First, the great Dr. Norman Pace and co-workers looked into this in a 2009 article in the Proceedings of the National Academy of Sciences seen here. Second, episode 72 of this podcast with Dr. Amit Pinto addresses water microbiology in a clear and interesting way. I'll put some links in the show notes to these things. And thank you, Carol. The hockey comment made me laugh. My Bio 350 Micronauts at the University of Puget Sound are currently engaged in a marine microbiological safari. My wife, Dr. Jennifer Quinn, and yours truly sampled seawater from the sites seen on this map of the Puget Sound area. Micronauts then plated out samples, dilution plated, on different kinds of marine media and looked very carefully indeed at the plates. Observation is key. First, of course, we look for luminous bacteria, as seen in this short movie. Next, we wanted to look for marine bacteria that broke down the agar that solidifies the plates, and saw multiple pits on some plates, seen here. Many of these pitters look fairly typical, like this one. Others, however, were strange and wonderful, like this spreading yellow one. The next goal is to restreak various candidates of interest for analysis. And as you can see from this weird restreak a student did, zooming in, strange things can be found. Folks on social media suspect activation of prophages or microbial interactions. It's all discovery, and I truly do enjoy leading these microbial expeditions so very much. Students learn so much for things that they find themselves. Most of all, as you can see, the Micronauts are fun to work with. I will miss teaching so much. Today's podcast guest appeared in episode number 15 of Matters Microbial. Dr. Sean Gibbons of the Institute of Systems Biology joins us again to discuss some of the subtle secrets he and his co-workers are learning about the human microbiome and how it impacts us all. I like to think of you, Sean, as the microbiome whisperer. In any event, welcome to the Quality Quorum again, Sean. It's great to see you, and I hope to visit your lab sometime soon.
Oh, Mark, you're welcome here anytime. We'd love to have you up, and it is a great honor to be on the podcast for a second time. Thank you for having me.
Oh, the microbiome is so interesting to people, and often it's used in a funny way. And so very quickly, for people who maybe didn't see your prior visit to the podcast, could you please explain what the microbiome is?
Yeah, so if you want to get technical with the language, the field usually refers to the sequencing data that we use to detect the microbes that live in, say, on the skin or in the gut or in the mouth, sequencing all these random pieces of DNA, mapping them to a database of taxa with known phylogeny, and like seeing, oh, you have viruses, you have bacteria, you have archaea, and that sort of detection of all those different organisms in the system via sequencing is what we often refer to as the microbiome. If we're referring to the actual living cells and organisms, we tend to say the microbiota.
I'm really glad that you did that because I am guilty of messing that up. And I'll make sure to put a link to that distinction in the show notes. So thank you for that.
Of course.
Your particular area that you like to look at, though, is some particular aspects of the human microbiome. This is correct? Yes.
Yeah, we've dabbled a bit in different biomes, but yeah, the human microbiome is largely what we look at.
I have to tell listeners and viewers there will be a link to Dr. Gibbons' website, which is really worth your time. Let me tell you, I have trouble with websites. This is pro, so you'll learn a lot from looking at it, and there will be a link in the show notes.
Thank you.
So, would you mind kind of giving and I don't know, this has come up a couple of times where the term thumbnail sketch comes from. Do people draw on their thumbs? I don't know. But could you give listeners and viewers just a thumbnail sketch of the research that you're interested in doing at the ISB?
Sure. Yeah. I mean, I guess I'm a systems biologist now that I work at the ISB. And, I've always been a microbial ecologist. I actually started off in the wet lab, growing things on plates. But, as a PhD student and a postdoc, I moved into computational biology, essentially. So I don't do many experiments these days. Our lab does a few, but not much. So most of what we do is collect data from largely human populations, but sometimes mice or other organisms, and try to understand, how the whole is greater than the sum of its parts, right? How do you take all these measurements from the different systems of a body and then stitch together those measurements to come out with some emergent understanding of how a given organism is contributing to disease or health or affecting the physiology of its host, or the host is affecting the physiology of the microbe. And so we like to dig into these sticky, complicated topics, not in simplified model systems, but going straight to the complicated system, right? Straight to the human gut. And usually it's sequencing.
Yes. And for listeners and viewers, it's really important to remember it is more diverse and numerous in the human gut than in the most complicated of rainforests. It is just remarkably huge and hard to sort out. And this is where computational biology was necessary. I have no idea how so much of what we take for granted today could have been done before computers got really good at that kind of thing. I should say programming, yes.
Yeah, I mean, biology became a big data field in the last 30, 40 years. With the advent of omics, we now can take thousands, millions of measurements from a single system. And that's, other fields have had the ability to do that kind of thing. Physics is one of them. They've been able to generate enormous amounts of data on the systems that they study. Astronomy is a great example, just training these really, sensitive cameras at the sky. And so we're collecting this incredibly rich data from the world, and within that data is embedded all of this complexity. And we've only, I think, what maybe we'll talk about today is we've only scratched the surface on understanding the underlying complexity of those data. We extract what we can from them, and we make some conclusions, or we get some insight. But there's still all of this residual information trapped in those data that haven't been touched. And so over the past several years, we've worked towards figuring out ways of reaching into that dark matter that hasn't been touched and extracting some new knowledge that we can add value to these existing data that are out there floating around.
So two things for listeners and viewers. A lot of microbiome data is publicly available for people to then analyze, which is a wonderful thing. The second thing is, and I know your father's a poet, and I don't mean to intrude on this at all, but I would say that we cast a shadow on our microbiome, and it casts a shadow on us. And because we're one and the same in the sense that we interact with one another. And the reason it's important for people to have at least some understanding of this business is that I see everywhere where commercial interests have begun talking about the microbiome. For example, a yogurt that they claim that has microbes that will reduce the incidence of depression. Sounds good to me, but I don't know the data for that. Additionally, there's a health clinic right next to my coffee shop that says they will improve your gut microbiome. And it's all I can do, Sean, to keep from going on and asking them questions, but I don't.
Oh yeah, there's a lot of claims out there being
Yeah.
made about this field. And there's a lot of fields like this. Anytime there's a new and exciting field of medicine that touches on human life, people are excited about it. I think the longevity space is like this right now too. There's all these hacks of supplements and things you can take to extend your life. Similarly, there's all these companies trying to sell you on personalized diets or supplements or probiotics with and there's a really exciting science going on in
Mm-hmm.
these areas, but they're trying to make leaps beyond where the current state of knowledge is to application, and we're getting close, I think, in these various fields. But the large randomized clinical trials that actually prove that this works for this indication, like much of that work hasn't yet been done,
Yeah.
and so a lot of that is just kind of smoke.
I want to be very clear. It might sound to listeners and viewers like I'm negative about these things. I am negative about some extreme claims is what I'm trying to say. And I'm sure it wasn't original with Carl Sagan, but the saying is extraordinary claims require extraordinary evidence. And I don't want to carry it too far, but what I like about the scientists I admire, and Sean, you're one of them, is you're very cautious about your statements, because if you just look in the media today, I saw an article today about one of the oldest people in the world who died at 117, and there's some work done on their microbiome, and the tagline is has a youthful microbiome, and that may be true, but it also may not be true because I haven't read the article and I'm just looking at the headline, and that's what most of us do.
Totally. Yeah, I saw that very same article. These N of one studies are interesting,
Yeah.
and they can be useful. But yeah, to make any broad claim from such a study, in fact, some of the work from our group has, I think we talked about this in our last episode,
Mm-hmm.
possibly, but we've shown that the microbiome of an older person who's healthy actually looks quite distinct from a younger
Yeah.
person's. And so
Mm-hmm.
you know, this centenarian might actually have been falling off a cliff in their microbiome, right? They may not have had a healthy microbiome, even though it
Right.
looked like a 20-year-old's. Yeah,
So I really appreciate that, because a lot of folks will think, microbiome, I'll read about that on CNN or whatever, and I'm not casting aspersions on CNN. I have to be so careful these days. I am saying that it's important to learn to talk about science and listen to scientists about these things, the folks who do the work, and they're cautious and careful. They're aware of proof, and that the plural of anecdote is not data, right? That kind of thing. So that brings me to one thing I'd like to ask you about and get your opinion on, if that's all right. As a college professor, and I'm looking for things that will stick in student minds about microbiology, it's very common to hear the statistic that for every human cell in me, there are 10, and they say bacteria, but they mean bacteria and archaea and presumably fungi and viruses and everything else. But it looks like, and I'll put links to this, that when folks tried to examine where that number came from, that everyone was quoting good, solid people, it was all based on a guess in the 1970s. And then there was actually no solid data on it. But then I look at a paper that comes out, Sean, about where you're actually counting the number of bacteria present. Because, listeners and viewers, not every microbe that is alive in and on you can grow on nutrient media. That's the issue.
yeah, biomass in the human body. How much of it's microbial? There's actually, I can't take credit for this. There's a paper from the early 2000s, kind of revising the estimate of the balance of biomass or the balance of cell number, right? Where we've kind of updated from like 1 to 10 to about 1 to 1, right? And
Yeah.
this is from, I think, PLOS Biology. I forget the authors' names. But they kind of revised the estimates. Of course, all of these estimates are pretty noisy because I do think that biomass can vary, you know, over time and across people even. But I do suspect that in the human gut, we maintain a fairly steady level of biomass. So what you're getting at in this paper that we just published is a method to kind of extract that information directly from sequencing data. There are groups out there trying to measure the number of cells that are in a gram of stool. They use flow cytometry or qPCR or other methods to try to, do an extra measurement to try to extract that information. And I'd always had a little bit of a weird feeling about some of these measurements. So to lay out my concern for the cytometry methods, and this is groups out of Europe that mostly develop these. They call these microbial load estimation. They take the wet weight of stool, so they take a few milligrams of stool, and they'll dilute that in some liquid medium, and then do flow cytometry to count the number of cells per gram of wet weight stool. And you look at those papers and you look at what are these biomass measurements associated with, and the strongest correlates with these biomass estimates are constipation and diarrhea. And so to me, that tells me that the measurement is completely conflated with water content. So they haven't measured the biomass per gram dry weight. They've measured it per gram wet weight. The dry weight measurements are harder to do, and they do it all the time in environmental microbiology. So the soil people, that's what they use. But in the gut, for whatever reason, everyone's using wet weight, and that's completely confounded with water content. And water content in bowel movement frequency or transit time, these can be somewhat decoupled from biomass. And so I suspect that they're not perfectly correlated biologically. And so what we wanted to do is have a measurement that was independent of this wet weight thing, but also a way to calculate biomass that was independent of having to do any additional measurement. So they could go back in time to, like you said, all of the publicly sequenced metagenomes that exist on SRA that you can download today. If you could just get a number, a biomass number for all of those, you could go back in time and ask, how does the amount of bacterial biomass in all of these samples correlate with human health or disease or this or that? So that was the basic task we put ahead of ourselves. And I can tell you how we did it, but maybe I'll stop for a second in case there's a follow-up.
Oh no, this is awesome. And I mean, for folks who, are thinking, well, we're having potty talk here. No, this is how we have to study a lot of things. And it's a monitor of what's in the gut, what passes through. And these are serious questions. Your physicians have questions very much like this that you have to deal with. So even though when I talk about these things with students, they kind of give me funny looks and we try and make jokes about it. But the fact is, this is serious business for some people in particular, heavily involved in health. So it's really important to look at. So I really like the fact that you're trying to find a technique independent method. And I guess collection independent method would be the way to describe it, of monitoring numbers. And I think that's great because this will give some idea of the power of this approach for other things you're going to talk about today.
Yes. Okay. So the way we did it is we had some interesting observations from some prior studies. So we had a mouse study where we treated the mice with antibiotics. And we were doing, at that time, amplicon sequencing, so just 16S, a marker gene of taxonomy. When we sequence these mice and you treat them with antibiotics, there's a big change in their microbiome. You see a big turnover, lots of stuff happening. And then one of the biggest signatures we found was that there's a huge enrichment in chloroplast and mitochondrial sequences, because when you amplify 16S, you get those plastids as well. And what that told me is that, you've crashed out all of the bacterial biomass. The primers we use for amplifying bacterial 16S aren't that efficient at amplifying chloroplasts and mitochondria. But if that's all you have left in the system, they're going to amplify those things. And so you see these host plastids become the majority of the signal. So I thought, oh, if you had somehow like a ratio of, if the host is just always there, it's just the bagpipe drone of the system, and the microbes are fluctuating on top of it, you could maybe use the ratio of those two as a measure of the total bacterial biomass in the system. And so we tried to test out whether that was an effective way to do it in metagenomes, because in metagenomes, you actually do get human DNA sequenced. Less than 1% of the total genome is human DNA, but it's in there. And so we thought if we took a metagenome and we annotated all the reads that are bacterial and all the reads that are human, and you just divided one by the other, and that it's a unitless number, so we can't actually say how many cells are there, right? So we're not, it's just this unitless number, but it is proportional to how much biomass is in the system, or we thought so at least. And so then how do you test that is the case? So we had some data sets where people had done these other historical measurements of biomass like qPCR or cytometry, and we could compare our method to those. We looked at cytometry first, and there was a correlation, very weak. It was very, very noisy. And if we looked at water content, the cytometry measurements were very strongly correlated with water content, where ours were not at all correlated with water content.
Good.
So then we moved on to qPCR, and qPCR was much more tightly correlated with our metric. And so for whatever reason, I think qPCR maybe is more independent of this water content issue. Kind of makes sense. And then finally, we had spike in data. So people can actually spike in an organism that's not in your system. Like if you're sequencing the human gut, maybe you take a deep sea microbe and you spike its DNA into your sample.
Sure.
And you can take the ratio of that, everything to that organism as sort of the biomass estimate for your system. There were some data sets that had done this, and so we could compare the host microbe to host ratio with this spike in ratio, and that showed this tightest of all correlations. They were almost the exact same number, and so it looked like it worked. It looked like it was doing pretty well. We did a few other validations, but I'll end off with a figure that I sent you, which was the final validation of the study we did. And we had data from a human trial where they had healthy human volunteers, and they gave them all a round of antibiotics. They took a few days of antibiotic treatment. And so when you do that, you can measure this bacterial to host read ratio, and you get a number that's, E to the 9.7 or something like that when you're before antibiotics. And then on antibiotics, they saw like a couple of order magnitude drop in this ratio. Makes sense. That's what we expected. And then a pretty fast recovery after that. So the first blue plot, the blue line is showing this bacterial to host read ratio in a human cohort with a transient dip due to antibiotic treatment.
Right.
And then recovery. The green plot is the same data set. We also wanted to see, well, are there other denominators that we could use to normalize? So we recently had a method that we can talk about it later to extract all the dietarily derived DNA from a metagenome. And if we divide the bacterial reads by the diet reads, you get the same answer as you get for the human. So if you normalize by diet, if you normalize by the host DNA, you seem to be able to get an accurate estimate of the total bacterial biomass. And then the final plot, which is a red line, this is from a mouse study where they treated a bunch of mice for many days, I think a week of antibiotics, and you can see they get an even bigger drop. I think they get a 2 or 3 order magnitude drop in biomass according to this ratio, and then a recovery over time. So this was our final validation for the method. There's still a lot of variability for the healthy folks. So even without antibiotic treatment, there's a fair amount of biomass variation. Whether that's just noise or whether that's real variation associated with diet or this or that, TBD. I think there's more work that needs to be done to validate this method. One of the caveats is, let's say you have inflammatory bowel disease and you have like a inflammatory event and you have more shedding of host tissue, the amount of host DNA is going to go up. But in the paper, we sort of argue that, well, in those situations, at least in the human gut, if there's a bunch of inflammation and a bunch of tissue damage, the gut becomes aerobic, all the strict anaerobes crash in their biomass, and the bacterial biomass will also crash in those scenarios. So whether it's the bacterial biomass going down or the host going up, I think they're kind of inextricably linked. So I think however you cut it, it's a pretty decent metric for assessing total biomass.
I think that's a wonderful idea. And I wanted to tell you parenthetically, when I was a teenager, long story, at that time, it was before Accutane, they gave me a treatment, well, several treatments for acne. And one of them, they put me, Sean, on a gram of tetracycline a day for about 7 years.
Oof.
Yeah. And I had to go in and get my blood tested once a month to make sure that my mitochondria weren't crashing. But the reason I tell you this is that when I took the microbial diversity course, there were 30% of the Bacteroides in my gut were still TET resistant after all that time. So the things that we do, like the shadows that I was talking about between the microbe, the macrobe, and now the environment, there's quite an interplay. And that's why the idea when you were on the program the last time, when you talked about precision medicine, you're also alluding to the fact that individuals are different from one another. So to tease out themes or trends is always a challenge. And it comes back to ecology and mathematics, which in some ways are the same field.
Completely. And it's this extra piece of information that was hiding in these data that we weren't using. We weren't leveraging this. And if you're looking at people who are sick or non-responders and responders to a drug, and you're just looking at the relative abundances of the bacteria, and in one set of patients, you see a complete collapse of the whole biomass of the system, but you're not taking that into account. I think, these shadows that are cast, we're all in Plato's cave looking at the shadows on the wall. But as we add these new ways of seeing into the data, we get a little more color, we get a little more depth, we get a little more insight into what's going on.
Well, you scratched my itch to learn a little bit about how you're measuring relative numbers, and that was important to me, so I could have some kind of not quite an estimate, but a qualitative feel for the number of microbes versus the number of okay, how much of Mark are Mark cells versus microbial cells? And this is an approach to actually do it that isn't dependent on the way in which one samples.
Exactly.
Now,
and
understand
you
that
don't need to make a new measurement.
Right. And understand that this approach that we're talking to is just one part of this huge assembly of organisms that I call me. Unless you're going to put me in a blender somehow. Let's not do that, at least not yet. So I think this is very interesting, and I'm really glad that you talked about it, because I think that leads very much in to how we can use this to start ask questions about health and even in some cases disease.
Yeah, and it just gives me a lot of satisfaction. A lot of the papers I've published over the years have been just reanalyzing existing data and squeezing a little more juice out of them than had been squeezed before, kind of using all the parts of the buffalo, right? And that sort of efficiency, it's like very cheap if you could totally run a lab that was just reanalyzing other people's data and coming up with clever little twists on how to extract more information. It's just really fun, and you have to be kind of clever, and you have to sit and think and look at the data for a long time, and wonder, like, why is there this little bias, or why is this little correlation going on? And it just leads to these fun little puzzles. So I definitely also write the big grants and try to get the big data sets, and I'll play the game. But this side of it, I think, is really fun. Just kind of extracting more value from data, which I think I have more examples of this if we want to kind of move to the next example. Great. So one thing that I was really excited about several years ago at this point was a paper with Tal Karem was the first author, I think, from Eran Siegel's lab, where they came up with a new metric you could get from a metagenome, which is called a peak to trough ratio. And what peak to trough ratios are a ratio of the mapping depth of short reads to the origin of replication versus the terminus in a reference genome. And so why would we want to measure this? So it turns out E. coli can split its cell like within like 15 or 20 minutes, but it takes something like 30 to 40 minutes for it to replicate its genome. And how does it square that circle? It parallelizes genome replication. And so you open up multiple replication forks that are going at once when you're growing really, really fast. And if you take that fast growing cell and you pop it open and you shatter its genome into tiny pieces, sequence it, and then map it back to a reference, you're going to get more reads mapping to the origin of replication and fewer to the terminus. And the slope of the line in that read depth mapping is proportional to the rate of replication of that organism's genome. And this is a beautifully elegant way to kind of measure this, right? And it was validated in vitro, so they could grow E. coli and chemostats at different rates and just see this nice relationship between the slope and the growth rate. And then they started looking at metagenomes and found that for the abundant organisms that are in a metagenome, where you have enough reads to map back to your reference, you can measure these peak to trough ratios in situ. It's like getting an instantaneous growth rate for a bug that's living in a sample as you got it. Really hard thing to measure by traditional tools, but you can just get this from data. So I always love this. I always love this way of measuring growth rates in real world samples. But I was also kind of puzzling over how to think about dynamics in the gut microbiome. In my postdoc, we had worked on time series analysis of daily poopers. So you're getting poop samples every day for people and sequencing them for like a year or 2 years, and then trying to understand something about what's going on in the system by analyzing those day-to-day dynamics. But there's also in the back of my mind, something I was uneasy about something. So now I'm going to go to this figure I gave on kind of the gut as a bioreactor, and this is from a paper from a couple years ago in Nature Communications. Joe Lim was the first author, and so what the figure is showing is kind of my very simplified model of the gut, right? So you eat food, and that food is devoid of gut microbes, hopefully, and it goes in and pretty much in the stomach and the small intestine, there's not a lot of bacteria. The host actually does its best to kill everything with acid and antimicrobial peptides because we want to have premier access to nutrients. But then once things get to the colon, that's an evolved organ. It's evolved to be an anaerobic fermenter for bacteria, essentially, an archaea. So when they hit the colon, all those nutrients suddenly get colonized by gut microbes, and you're off to the races, and everything starts growing. So biomass starts to rapidly increase in the colon from the top to the bottom of the colon, and then you poop, and everything falls out of the system. So it's kind of a semi-continuous bioreactor where you get these boluses of material moving through that are colonized and accumulate bacterial biomass and then are excreted. And in the plot, you see these growth curves for taxon 1, taxon 2, taxon 3. And for taxon 3, it actually achieves its carrying capacity before you poop. So it's kind of stopped growing, it's slowed down, but taxon 1 is still ramping up. It's still just getting going. And you poop and it's still growing as fast as it can grow. I don't know if this is what's actually happening, but are organisms actively growing when you poop, or are they all sort of senesced and stopped growing and in stationary phase? At the end of the day, what we measure are these day-to-day poops, which is just a snapshot of the end point of all those internal dynamics. We can't actually measure growth rates of bacteria from daily poops because they're growing with doubling times of like 20 minutes to a few hours. Mm-hmm. But people were fitting lockable Terra models, which assume that you're able to get growth rate estimates from the fluctuations day to day, which I think is incorrect. So when I was analyzing these fluctuations, you saw these really interesting patterns, like your change in abundance from day 1 to day 2 is negatively correlated with your abundance on day 1, and you get your deltas essentially cross 0. So if you are below your average abundance, you tend to have a positive excursion in your abundance at the next time step. And if you're above your average abundance, you tend to go down at the next time step. And what this looked like to me was regression to the mean. If you're just sampling from a Gaussian distribution, if you sample far from the mean, you're likely at the next sampling to sample closer to the mean. So it sort of pointed to these bugs are maintaining an average carrying capacity in the system, that they're growing, our diets are fairly stable and we poop at a fairly steady clip. And so every day you're sampling a bug at its sort of carrying capacity or wherever it gets to in its growth phase, at that amount of duration in your gut. And that's relatively stable day to day because these average abundances that we measure are very steady over months to years. People maintain very stable average abundances of all of these taxa. Okay, so that's the start of the journey for this particular paper. Now I bring in the peak to trough ratios, which I was talking about before, because we can also get time series of these peak to trough ratios. So not just look at the changes in abundance, but look at fluctuations in these growth rate estimates of bugs over time. And I mentioned that fluctuations in abundance and baseline abundances are always anti-correlated with each other. Like there's some sort of regression to the mean. But when you look at the abundances versus the peak to trough ratios, the relationships were all over the map. Sometimes they were positively associated. Sometimes they were negatively associated. Sometimes there was no association. There's all this richness to those associations that was unexplained. So the way we thought about this, and so Joe Lim, the first author, he has a master's in applied mathematics. So we were thinking in terms of equations. So now I'll move to the next figure, which is the growth phase estimation kind of cartoon. If you think about growth of a bacterium, the simplest way you can formalize growth would be something like a logistic growth equation, which is that sigmoid curve, an S curve, where you're starting at very low biomass and you grow exponentially, and then you taper that growth and hit some carrying capacity. And it's this dx dt is equal to r times x at t multiplied by 1 minus x of t divided by k, which is the carrying capacity. So that's the equation. It's not too complicated. And so I was staring at this S curve and thinking about, okay, how can we explain these varying relationships between peak to trough ratios and abundance over time? We took the derivative of the S curve, and that just gives you this unimodal peak, right? The growth rate peaks in the middle of that S curve. And then we took the second derivative. So we're just playing around with the math here. And what really got me thinking about this was the second derivative. So in the second derivative, at the beginning of the growth curve, there's a positive acceleration in the growth. And in the later part of the growth curve, there's a deceleration. Right? This is kind of obvious in hindsight, but it just got the brain working on it. And so then, it stands to reason that, let's say I have a person who's a fast pooper, there's a bug that's living in their gut and they have diarrhea. And so it doesn't have a lot of time to grow. It's got less time to achieve its growth curve, point in its growth curve. So it's going to tend to be sampled earlier in its growth curve. And because that person has a fairly steady bowel movement frequency, it doesn't vary that much day to day, you're going to kind of noisily sample within some region of the growth curve in that fast pooper. A slow pooper, they're going to let that bug grow for longer. The poop is sticking around for longer in the system. The bug has more time to achieve its carrying capacity. It's going to get closer to that end of the growth curve. And then you sample it, kind of jittered around close to that point. And then you plot the relationship between the growth rate and the abundance. In the acceleration phase of the curve, it's a positive relationship. In the deceleration phase of the curve, it's an anti-correlation. They're negatively associated. You're increasing your growth rate and your abundance in the beginning. In the end, you're still going up in abundance, but your growth rate is decelerating. And this would elegantly explain these variable relationships that we observe. So then we applied this to data, and we found that for the only 4 people that we had these really long, dense metagenomic time series for, 2 of them pooped faster than the other 2. So 2 of them were pooping twice a day, and the other 2 were pooping once a day. And if you looked at the estimates of what growth phase their microbes were in, there was an enrichment in things that should be in the earlier growth phase, in the fast faster poopers and
Right.
an enrichment in things in later growth phase and the slower poopers.
That makes
We
sense.
want to
Yeah.
test, yeah, we want to test this in like more time series, but
Sure.
anyway, we think it's a way to estimate the in situ growth phase of bacteria, was previously inaccessible to us. And what it tells us also is that most things in the gut are growing actively when you poop. The things haven't stopped growing. You poop and they're still going like gangbusters.
That's really interesting because, again, people make assumptions. And when you think about stuff that's resident in this bioreactor, you don't think of it as being, I'm not going to say it's all in stationary phase. And I have thoughts on that subject too. But that's a really interesting way of measuring. Most people would say, well, how do you measure growth rate? And you look at through the microscope and you do optical densities and you do things of that nature. But what you're doing is using this relationship to the terminus to the origin of replication that gives you a culture independent technique for doing it. And in addition, if I'm understanding you correctly, you can fish out that data for any given taxon that you want to look at that's fairly abundant. And that's power.
Right. I mean, and one thing is if you don't have a time series, we had some in vitro data from E. coli, and if when the bugs hit stationary phase, their peak to trough ratio just drops to nothing. But as long as they're growing to some extent, it's very bimodal. The separation is very clear. So if you just had a
Wow.
snapshot metagenome, you could kind of categorize bugs into these are active, these are not active, just based on that measure. But if you
Wow.
had time series, you could even categorize whether they were early or late in their growth phase.
I got to tell you, I'm teaching some aspects of this in just a few days because I talk a lot about growth rate and I talk about Vibrio nutrenges that can divide every, I believe it's 11 minutes, but it takes about 25 to replicate its DNA, right? And so your technique is independent of any other technique for growing it. And so that's what I really like about this. Wow.
Yeah, and some of the bugs in the gut we actually don't know how to grow yet. And
Mm-hmm.
so, you can get this kind of information from things that we can't culture.
And it'll give you information as some of the people have been they're believers in what I call angel microbes. And so you'll hear this particular microbe is the best microbe to have for optimal health. And I see ads for this, and I'm not going to name any of the ones that they kind of suspect, and you can if you'd like. But you can actually ask the question, is it actively growing? Is it stationary or senescent or whatever term you want to use? You can answer that question even if you can't grow the organism. And if you could grow the organism, that doesn't tell you what it's doing in the gut.
Exactly. Although one really simple metric that seems to work is if it's abundant in stool, it's actively growing. Like it's impossible not to be. I think just it just sort of has to be the case because in order to achieve that amount of biomass relative to everything else that's in
Yeah.
there, you had to have been growing very, very fast.
Now, in other contexts, Sean, and you've heard me talk about this, sometimes I get, when I say suspicious, there's no value judgment about using stool samples because I don't know, and I hate to talk about the geometry of stool, but if we're interested in the organisms that are actually adhering to the gut epithelia, how on earth would you sample those? So, we use that as kind of a monitor for what's going on. But I think as the technology improves, we'll be able to evaluate things in a way that we never have before. And sometimes, as you've mentioned, from data that's publicly available.
Or perhaps, or samples and biobanks that could be
Yes.
reprocessed in some new
Yeah.
way. Yeah,
Well,
I like stool. I think it's a pretty good proxy, but it's true that if you sample the tissue, you will get different bugs enriched at the boundary. But that mucus layer is getting massaged in and sloughed into the stool constantly as well.
I completely agree. What I have seen in an animal system with which I've worked is that if you wait before you collect that stool sample, there's a bloom that's taking place. So
Oh yeah,
you've got to know when you're collecting and be consistent about it. And when I have that conversation, people who aren't scientists kind of roll their eyes, but it makes perfect sense because all joking aside, there's a lot of nutrients that are in the process of still being used. So that's an opportunity for some microbes, even after you have offloaded that particular part of their biomass.
I mean, part of our lab, well, the one experimental system we have are poop soups, where we take fresh stool from people, mix them up into PBS buffer, and then cultivate them in the anaerobic chamber. And those poop soups are bubbling. They are
Yeah.
active. Like there's still food in that stool that they're eating,
Mm-hmm.
and they're growing. Yeah.
Yeah. Anyway, what wonderful stuff. And does this not lead into the dietary issues that you were going to talk about now?
Yes. So the triptych of new ways you can extract information from existing data. Um, we recently published a paper to see, ask the hubristic question, can we actually tell what you're eating just by sequencing, shotgun sequencing the DNA from a stool sample? Um, so maybe the average person doesn't realize how hard that would be, but if you looked at the amount of DNA coming from the diet that's in stool, it's minuscule. The vast majority of the DNA in stool is bacterial, and then archaeal and viral. There's a little bit of human DNA, and then it's like 0.001% of the DNA is coming from dietary substrates. So not a lot survives passage, and and that leads to a lot of issues with how do you extract such a low frequency signal from this enormous mishmash of stuff? It's a needle in a haystack kind of problem. And other groups have thought about this a lot. So historically, in humans historically, we collect dietary information through questionnaires because we can ask people what they're eating, which is great and it works pretty darn good. But it's not perfect because people lie about what candy bars they ate and this and that. Um, but in systems where you can't ask questions, you need something else to work. And so in the wildlife field, in kind of wildlife microbiology, microbiome field, for a long time, they've been using amplicon sequencing of primers that target chloroplasts or mitochondria on stool from gazelles and grizzly bears and wild animals to be able to get a handle on what their diets are. That's where all the innovation initially came from in this poop-based diet detection. And the lab that took this and ran on the human side has been Lawrence David, who's at Duke. And so his lab has developed a database of chloroplast marker gene sequences from a bunch of things in the human diet. He's perfected these primers. I believe he's also working on mitochondrial primers. And he has a method called FoodSeek, where they use amplicon sequencing to take this needle in a haystack and use PCR to amplify it so you can actually get enough to sequence, which is great. So Christian and myself, so Christian was a senior research scientist in my group, and now he runs his own lab at the Medical University of Graz in Austria. We were thinking, is it possible to just take a metagenome and get this same kind of information? How good or how bad would it be? The nice thing about it is you don't need to do any extra steps experimentally. You don't need to run PCR. You just take the existing metagenome, you run it through the pipeline, and you get an answer. It took 5 years to get this to work properly. And I'll say some of the tea behind getting it to work properly is there was this conversation between the Salzburg lab and the Knight lab with the cancer microbiome thing, right? That there was this kind of signal of being able to detect different cancers by the microbes that are present in blood or tissue. And it turns out that a lot of those signals were artifacts, right, of batch effects and processing issues. And the bioinformatics wasn't properly adjusted. And no one is to blame for any of that, right? Any of us could have made these mistakes. But the conversation between these two labs was very productive because it gave us essentially a perfect roadmap for how to design our bioinformatic pipeline to avoid these very issues that came up. So what are the things we did? So one thing that we did that wasn't done in that original cancer paper, that original cancer paper, they took a metagenome and they mapped ostensibly all of the human reads to a reference genome with kind of an alignment mapper like Bowtie2. So they thought they extracted all the human reads from the sample doing that. And then they took that and ran it through Kraken 2 And the Kraken-2 database did not contain the human genome because they thought they had gotten rid of all the human reads. Well, it turns out even if you map to a reference, there's still a bunch of human reads that stick around. They're not all mapped away. This is actually relevant to that first story I told with biomass ratios of bacteria to human reads.
Right.
One of the things the reviewers asked was, a lot of metagenomes that are uploaded have had the human reads stripped away with pre-mapping. We tested this and found, well, yeah, sure, you see an order of magnitude drop when people do this read mapping, but there's still a bunch of human reads there. And weirdly enough, even after mapping human reads away, our biomass estimate is still quantitatively accurate. The samples with high ratios and low ratios are preserved before and after this mapping process. But anyway, that's an aside. Back to the cancer paper. Because they didn't include the human genome in the Kraken database, there were still these residual reads that would then get placed into other bins by the classifier and led to spurious signals. It turns out in the human genome, we have mitochondrial DNA, and sometimes there's contamination in reference genomes, and this can lead to sort of off characterization or off annotation of a given read. And so you got a bunch of artifacts. So the first thing we did was include a bunch of decoy genomes in our database. So we had the human genome, we had hundreds of genomes from all these plants and animals and fungi that are in the human diet, in addition to every microbial genome from RefSeq, all the archaea, all the viruses, all the plasmid sequences. So we just made this big gargantuan 5 terabyte reference genome database that we pushed through Kraken-2. And having all of those references really depressed the amount of false positives that we were getting. And then the second, I think, big thing that we did that made it work was to include an entropy filter. So let's see if I can explain this. So any given read can get multiple classifications. You can break it into k-mers, feed that into Kraken-2. And some of those k-mers are going to look like they came from a banana tree, and some of them might look like they came from the human genome, and some might come from a bacterium. Those reads are not informative. They are taxonomically useless. Traditionally, though, Kraken will take the winner, the number of annotations that sum up to the largest number. That's what it calls it. So it's a banana, right? Even though it has all these other annotations embedded in it. And so we run an entropy filter to look at the phylogenetic tree and ask, what is the diversity of classifications of this read? If they're too disparate across the tree, we throw that read out and we get rid of it because it's not useful. And between that and the decoys, we get almost no false positives in our method. So
I like how you're checking your work, and this is really interesting because I'm watching you speak the language that, for example, my wife speaks of mathematics, and then coming back to the actual biological issues as well. So folks who are thinking about becoming scientists, it's important to be conversant in both languages.
Totally. Especially in the big data era of biology. But I guess I should talk about some more of these figures that I sent along. So the one is this phylogenetic tree. It's a big circle plot, and it shows all the different food-based taxa that are in our database. So we have something like 400, and I forget exactly how many, over 400 reference genomes from plants, animals, and fungi that are in the human diet. And on the tree, most of them are complete genomes, but there's a part of the tree that's kind of wonky looking with long branch lengths and stuff. These are assemblies, so they aren't actually complete genomes. They're just sort of partial assemblies of these organisms that we still added those to our database. There's some heat along the edge of the tree with different colors. And this is a little value addition we did for this method. We tied our reference database of the taxa to FoodDB, which is a database of the metabolic content of the biomass of these different organisms. So if you take a gram of beef, how much cysteine and methionine and this and that and the other are in that beef. So we can get a nutritional content of each one of these organisms. And so on the tree, you can see there's kind of a redder edge, which are the plants. There's more purple down towards the middle center, and those are the animals. They're enriched in protein. And then red again for the fungi, they're more carbohydrate enriched. The black bars on the very tips of the tree are the energy content per gram of that biomass. So on the plants, you see big jumps for like seeds, have a
Mm-hmm.
lot of energy density. Animals tend to have a lot of energy density. And so this is sort of just an overview of all the taxa and their sort of nutritional content that are present in the database. And this is included in this Kraken database to be able to kind of pull these annotations from the very, very small fraction of reads that are coming from these organisms in a metagenome. Anyway, that's the database. We validated it in a lot of ways. You could read the paper if you want all the details. We had some controlled feeding studies. There was actually a really fun one where Hannah Holscher's lab at UIUC had a avocado study. They had two populations of people. In one group, they were all eating the same diet. In one group, they did not get a large Hass avocado for lunch. In the other group, they did get that large Hass avocado. This was paid for by Big Avocado. They wanted to look at the health effects of avocados. And when we run their samples through Medi, there was only one thing that was significantly different between the groups: avocado. It was very, very significant, and was the only thing we detected that was different between the diets. So that was a great validation, and we had more as well. One of the final things we did is the final plot that I sent you, which is to take a data set where we didn't have any dietary information. This was the Metacartus cohort. This is a European cohort that was recruited to study cardiometabolic disease and the microbiome and the metabolome. And in that cohort, they had like people who were metabolically healthy, and there was a group of people who had metabolic syndrome. We didn't have any questionnaire data on what they were eating. We ran their metagenomes through Medi, and the left-hand plot is showing the fold change in the abundances of different organisms detected in their stool at the DNA level. And so you'll see on the top, sus scrofa, which is the Latin name for pig or wild boar. It was enriched in the people who had metabolic syndrome. These are also Europeans, I should state. So I don't know, there's maybe more pork consumption in like Austria and different parts of Europe. And in the metabolically healthy people, there is an enrichment in fruits, nuts, and vegetables, essentially. So if you were healthy in the healthy cohort, you tended to eat more fruits and veggies. And if you were in the metabolic syndrome side, you tended to eat more meat, which is fairly consistent with what we know about nutrition.
Yeah.
Um, we could take those same organisms and we could break them down into their nutritional content and ask, what nutrients are enriched or depleted in these different groups. And we found that in metabolic syndrome, as you might expect, based on the pig signal, there was more cholesterol. There was more omega-6 fatty acids like arachidonic acid. There was some stuff from milk, like lactose kind of stuff. And then in the healthy folks, there were more sugars that were detected. And
Hmm.
this is kind of counterintuitive, but our method doesn't understand processed foods, right?
Yeah.
It assumes everything is a whole food. When it sees a piece of DNA, it's like, oh, that's coming from a whole apple, not applesauce or something like that. And so the people who ate more fruit were inferred to have more sugar because fruit is naturally the most enriched in sugars in a whole food diet. Yeah, so we think it kind of works, and we're excited for people to apply it to all these retrospective data sets that are out there where, in a cancer immunotherapy study, you had people who didn't respond. Maybe they were eating different things, but they didn't collect that data. But now we can go back and look and see if that's the case.
So we're all very reductionist in our point of view, though that's starting to change. And systems biology is leading the way, incidentally. But this idea of looking at one cause for everything, people really want that to be true, and it just isn't. Also, as I'm watching you talk about this, I'm thinking to myself, if I cheat on my diet, my metagenome will tell.
Yes,
So there you go. A new
that's right.
worry. The
We see
metagenome is
this.
big
No, we see this all the
metagenome.
time.
Yeah. Big metagenome is watching, right?
Or omics, right? Like we have blood metabolomics from a lot of people, and there's this one molecule that shows up in the blood if you're a smoker, And there were several people in our cohort that claimed not to be smokers, but it was clear from their blood metabolomics that they were smokers.
Oh my goodness.
You can't hide
Well,
from
what
data.
Well, what I really like about this is we can talk about kind of the mathematical way of comparing and looking at things, and you led us on a wonderful journey where we saw how you could start to use archived data to determine how many what the bioload is, even though I know I'm using a term that is out of favor. But we can actually use that to answer questions. You went on and told us how we could even tell who was active and who wasn't. Again, not by culturing, not by measuring cell number. And now we're learning about how we can use this technique to determine diets from people objectively, right? And it shows the power of the approach in ways I had never expected.
It just shows how much mystery there is in these data sets that we haven't touched, and just a little bit of thinking about what could be there that we haven't seen yet. It's like the people who figured out with discovering planets with the Kepler satellite. If a little bit of dimming of a star, a planet passing in front of the star, you can learn all this information about the size of the planet and the atmosphere of the planet. Just that was in the data that was there back in the 1920s when people were taking these pictures of
Yeah.
the sky. But until someone thought about, oh, what if we looked at it this way? Suddenly, this whole universe opens up to us.
And again, this is because it's everything. Everything is complex, but that shouldn't stop us from looking and learning. And the term the dark microbial matter that people talk about, I would want to call this like the dark data, except that has been used by people with whom I don't have common cause. So maybe we should have you can't say cryptic data, can you? Right? Because you're looking
the mystery.
Yeah. You're not looking at Sasquatch genomes. So there you go. So I really have appreciated what you have brought to the table, the metaphorical table here. By the way, the idea, and I'm sorry, when you talk about Plato's microbial cave, I will be using that. That's wonderful. Do you have like 3 take-home lessons you'd like to give listeners and viewers before we close out?
Yeah, let's see. One is we live in a pretty cool time. If you're a kid living in a low to middle income country, doesn't have a lot of access to resources, but you have an internet connection and a laptop, you can do science and you can publish papers, right? Like, as you saw, a lot of the things that we did, there was no collecting any data for most of it. We just pulled down public data, looked at it a little bit, squeezed it, pushed it around, and came up with a way to put together a story that was useful. So I think you can, for a very low cost, for a very low overhead, anybody can be a scientist today. It's just about having those basic skills to start having access to being able to do this kind of work. But there are free courses out there. There were tutorials. There's even large language models that help a little bit with the coding, although be careful there because they hallucinate and make a lot of mistakes. But we're living in a very democratic era of anybody can do this. You don't have to be a big fancy scientist with a huge budget and a big grant. You can do science on a shoestring budget. And I would also that's also a message for my colleagues who are out there having their grants frozen by the NIH, and the Trump administration has been a horrible bane on science in the United States. And we should speak out against that and push against this autocracy that's happening in our country. But people who are squeezed by what's happening, this anti-science push, they can still do science on a shoestring budget. It can be done. So there's all this mystery, there's all this unmeasured stuff, all these things we haven't quite looked at yet in a certain twist of the data. And this takes us back, another take home maybe is, let's think back to the early days of molecular biology. These people were doing really sophisticated, interesting experiments with not much technology. Mm-hmm. They were using pretty simple setups, and they were able to extract really interesting insight. It just requires us to be creative, clever, and just meditate on the data, think about the data. You don't have to spend $1 million to do great science.
I think those are wonderful lessons. There used to be an essay, I'm sure you read it, It might have been John Roth, but no, it was John Beckwith, Toothpicks and Logic. And that's how he did a lot of microbial genetics, which is just amazing to read. I really want to thank you for this session, Sean. It means a lot to me. You've come on twice, and I've learned a ton. So I am really grateful.
Well, I am honored, Mark, and you are welcome up here anytime to visit.
I'm so looking forward to it. I hope you and yours have a great day.
Same to you.
Thank you. This has been Matters Microbial, a weekly podcast about the wonders of our microbial world and the people who study it. You can send questions, suggestions, or comments to me at mattersmicrobial@gmail.com. Show notes from today's episode, with, as always, epic links, can be found at microbe.tv/mm. If you like our work, please consider supporting us at microbe.tv/contribute. I'm Doc Martin, and you can find me in the biology department at the University of Puget Sound in Tacoma, Washington. Dr. Sean Gibbons is Associate Professor at the Institute for Systems Biology in lovely Seattle, Washington. Many, many thanks, as always, to David Renata for superb editing and Rebert Clark for the wonderfully quirky music. I hope that you've all enjoyed being part of our Quality Quorum for today. See you next time on Matters Microbial.